Now, yesterday we discussed the LAQs and some SIQs. Krebs cycle, glycolysis, and ATP balance sheet we discussed yesterday, right? Now, today we discuss the some SIQs, RQ values, and then ambibolic pathway, these two SIQs, and then LAQ C3 cycle. First, ambibolic pathway. Before ambibolic pathway, we discuss the respiration quotient. Respiration quotient. Respiration quotient. Okay. What is the respiration quotient? What is the respiration quotient? Volume of CO2 evolved by volume of O2 consumed. Volume of CO2 evolved by volume of CO2 consumed is called what? Respiration quotient. Okay. So in short form, it's called RQ. In short form, it's called what? RQ. So what is the RQ? What is the RQ? RQ means nothing but R means respiration, Q means quotient. Okay. What is the RQ? Volume of CO2 evolved by volume of CO2 evolved by volume of O2 consumed. Volume of O2 consumed is called what? A respiration quotient. If we are take the sugars, nothing but carbohydrates, what is the value of that RQ? And if we are take the fats, now what is the RQ of fats? Now we are take the proteins. Now what is the RQ of proteins? Now we discussed one by one. First we discuss the if we are take the glucose. If we are take the glucose. Now what is the glucose RQ value? Now if we are take the fats. Okay, what is the RQ value of fats? If we are take the proteins. Okay, if we are take the proteins, now what is the RQ value of protein? Now glucose, fats and proteins are substrate, respiratory substrates. These are what? Respiratory substrate. Respiratory substrates. Okay, respiratory substrates. If we are take the respiratory substrate as a glucose, then what is the RQ value? If we are take the respiratory substrate as a fats, now what is the RQ value of fat? Then if we are take the protein as a respiratory substrate, okay, now what is the RQ value? Okay, now first one, glucose. So what is the formula of the glucose? C6H12O6. This glucose is oxidized in the presence of 6 molecules of oxygen. 6 molecules of oxygen and release the what? Release the 6 molecules of carbon dioxide and 6 molecules of H2O, 6 molecules of either electron. No places here. So here 6 molecules of water and some amount of energy is released. Some amount of energy is released. Okay. Now what is the RQ formula? RQ equal to volume of CO2 evolved. How much CO2 is evolved? Evolved is nothing but release. How much CO2 is evolved? 6 CO2. 6 CO2. Then what is the formula? Then volume of CO2 evolved by volume of O2 consumed. How much O2 is consumed? 6 O2 is consumed. 6 O2 is consumed. So 6 1s are 6. Okay. Now volume of glucose RQ value. RQ value of glucose. RQ value of glucose is what? How much? 1. Okay. Is a 1. Okay. Then if we are take the fats. If we are take the fats. Now example of the fats is tripalmitic acid. Tripalmitic acid. For fat example, we take the one example of the fats. These are example tripalmitic acid. Okay. Now here carbon 51 molecules of carbon, hydrogen 98, and then oxygen is 6 oxygen. Okay, these are two molecules of tripalmitic acid. Okay, tripalmitic acid. Okay, these are oxidized in the presence of oxygen. Oxygen, how much molecules of oxygen? 145 molecules of oxygen, and release the water 102 molecules of carbon dioxide plus 98 molecules of water molecule, and then some amount of energy is released. Now apply to the what is this RQ formula? RQ. So what is the RQ? Volume of O2 CO2 evolved by volume of O2 consumed. So how much CO2 is evolved? Evolved means nothing but released. How much CO2 is released? 102 CO2 is released. 102 CO2 molecules are released. And how many molecules of oxygen is consumed? 145 molecules of oxygen consumed. So it's what their value is 0.7. 0.7. Then now means these are what? Less than the RQ value of tripalmitic acid or fat is less than 1. Okay. RQ value of fats are less than 1. Mean less than what? Glucose. Then now proteins. Now protein RQ value is directly protein RQ value is 0.9. Okay, so 0.9 in the proteins, RQ value 0.9. In proteins, RQ value of 0.7. In fats, RQ value of 1 in the starch, nothing but glucose. Starch is different. Starch is a polysaccharide, glucose is a monosaccharide. Okay, now RQ values of glucose. RQ value of glucose is 1. Then RQ value of fats. RQ value of fats is 0.7. RQ value of protein. RQ value of proteins is 0.9. These are important for 4 marks. Okay. Then now next amphiboolic pathway. Next is amphiboolic pathway. First, what is the amphiboolic pathway? Okay, why it's called amphiboolic pathway? Before that, we have what small basic. Amphi means both type of metabolism is occurs. Amphi means both type of metabolism is occurs in that step. Okay, first one is catabolism. Second one is what? Anabolism. You know, catabolism already we learn in the living world first year. Okay, catabolism means which molecules are breakdown. Okay, complex molecules are breakdown and converted to what simple molecules are called what a catabolism. Catabolism means complex molecules are breakdown to what a simple molecules are called what a catabolism, are also called, it's a destructive process, means breakdown process. Then next one is what a anabolism. Anabolism means simple molecules are converted to what a complex molecules. These are what a constructive process, means it's make the what a simple to complex. Okay, now means both in the amphiboolic pathway, this both type of metabolism is occurs. Catabolism means destructive process is there, and anabolism means it's a making process. Okay, it's a constructive process there. Constructive and destructive both process occurs in this step. That's why it's called what a amphiboolic pathway. Okay, what is the amphiboolic pathway? Amphiboolic pathway means both type of metabolism, constructive and destructive, both type of metabolism is occurs. It's called what a amphiboolic pathway. Amphiboolic pathway. Amphiboolic pathway. Amphiboolic pathway means both type of metabolism. Both type of metabolism. Both type of metabolism is occurs. Is occurs. Okay. What both type? Catabolism. Catabolism and anabolism. Catabolism and anabolism. Okay. Catabolism means what happened in the catabolism? It's a breakdown process. It's a breakdown process. Okay, it's a breakdown process, or also called destructive process. Destructive process. Destructive process. Okay, means what happened in the catabolism? Complex molecules are breakdown. Complex molecules are breakdown. Complex molecules are breakdown into simple molecules. Complex molecules are breakdown into simple molecules. These are what a catabolism. Then anabolism means these are constructive process. Constructive. These are what a constructive process or making process, or also called making process. Okay. What happened in the anabolism? What happened in the anabolism? Simple molecules convert into what a complex molecule. Convert into complex. Simple molecules are converted into what a complex molecule. These are opposite to the what a catabolism, right? In the catabolism, complex converted to a simple. In the anabolism, simple converted to a complex. Both are what opposite, right? So both the type of metabolism is occurs in where? In the amphibolic. Okay, right now, if we are take the substrate, respiratory substrate as a proteins, as a fats, as a carbohydrates, what happened? Now we discuss that one. Okay, if we are take the water, we are intake the what? If you are intake as a if we are take the water, carbohydrates, proteins, and fats as a respiratory substrate. As a respiratory substrate. Respiratory substrate. Okay. What happened? Now we discuss this. Okay. Carbohydrates. Carbohydrate is a polysaccharide. This polysaccharide is converted to what? Disaccharide like sucrose. Polysaccharide is converted to what? Disaccharide like sucrose. Carbohydrate is a polysaccharide. These are what? A disaccharide. Disaccharide. Di means two. Saccharide means sugar units. Okay. Then sucrose is converted to glucose. Glucose is a monosaccharide. Now glucose form enter in the where? In the respiratory pathway. Now glucose is converted into what? A glucose 6-phosphate. Glucose is converted to what? A glucose 6-phosphate. Okay. Then dash. You know already glucose 6-phosphate is converted to fructose 6-phosphate. Fructose 6-phosphate is converted to what? Fructose 1,6-phosphate. Directly लिख रहा हूं मैं. Fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate. Okay. This fructose 1,6-bisphosphate, acetylase class. Fructose 1,6-bisphosphate in the presence of what? Aldose or aldolose. Aldose enzyme. Its breakdown into what? DHAP means dihydroxyacetone phosphate. Dihydroxyacetone phosphate. Dihydroxyacetone phosphate, comma, sorry, bracket close. Okay. D for di, H for hydroxy, A for acetone, P for phosphate. Then glyceraldehyde 3-phosphate. Then glyceraldehyde 3-phosphate. Okay. Then this glyceraldehyde 3-phosphate undergoes several reaction and form a what? A pyruvic acid. Pyruvic acid. Okay. This pyruvic acid form the what? Acetyl-CoA. Okay. Acetyl-CoA. And next step is what? A Krebs cycle. Okay. Krebs cycle. In Krebs cycle, release the water, carbon dioxide, and a water molecule is released. Carbon dioxide and a water molecule is released in the Krebs cycle. If you are take the water, glucose, this glucose is directly enter in the respiratory pathway. Means this 6-carbon compound of glucose partially break down into the water. 3-carbon compound here, 3-carbon compound, then pyruvic acid 3-carbon compound, then acetyl-CoA 2-carbon compound, and then finally carbon dioxide is released. Okay, means glucose is breakdown into the water. Glucose molecules completely break down and release the water in the form of carbon dioxide. If we are take the glucose as a respiratory substrate, okay. If you are take the water respiratory substrate as a glucose, what happened is this glucose is directly involved in the respiratory substrate and released in the form of carbon dioxide after complete breakdown. Okay, then if you are take the fats as a respiratory substrate, what happened? If we are take the water fats as a respiratory substrate, what happened? First, these fats are break down into the water fatty acid and glycerol. Fats are break down into the water fatty acid and glycerol. Fatty acid and glycerol. Okay, now this glycerol. This means fats are not directly enter in the respiratory pathway. Fats are break down to fatty acid and glycerol. Now this glycerol is converted to DHAP, then enter in the in the respiratory pathway. Then fatty acid is converted into what? Acetyl-CoA. Fatty acid is converted into what? Acetyl-CoA, then enter in the respiratory pathway. Means fats are not directly enter in the respiratory pathway. Fats break down into what? Fatty acid and glycerol. Glycerol in the form of DHAP. Glycerol in the form of DHAP means glycerol is converted to DHAP, then enter in the respiratory pathway. And fatty acid also does not directly enter in the respiratory pathway. This fatty acid also converted to what? Acetyl-CoA, then enter in the what? Respiratory pathway. Okay. Then if you are take the proteins, if you are take the what? Proteins as a respiratory substrate, what happened? Proteins as a respiratory substrate. Proteins. If you are take the water proteins as a respiratory substrate. Proteins. Okay. Now proteins are breakdown. Proteins are breakdown into what? Amino acid. Proteins are breakdown into what? Amino acid. See, this amino acid also does not directly enter in the respiratory pathway. This amino acid, it's convert into the water. Maybe enter the water pyruvic acid, or also maybe water acetyl-CoA, or also maybe water directly enter in the Krebs cycle. Does not directly enter in the amino acid form. It's convert in pyruvic acid, 3-carbon compound pyruvic acid, or 2-carbon compound acetyl-CoA, or indirectly enter in the Krebs cycle. Mean what here? Fats are breakdown, convert fatty acid and glycerol. Breakdown means which process? Destructive process. Okay, here glucose also breakdown. Okay, glucose is breakdown to what? Step by step. Okay, these are also called a destructive process. Here protein is a polymers of amino acid. So proteins also break down into the what? Amino acid. Okay, so these steps are a breakdown process. This step is what? Fats are breakdown, carbohydrate nothing but glucose are breakdown, proteins are breakdown. So breakdown is nothing but which process? Breakdown is nothing but which process? Breakdown is nothing but catabolism, means destructive process. Then if our body required the fats, if our body required the fat, what happened? This acetyl-CoA is withdrawn. This acetyl-CoA is withdrawn from the respiratory pathway. Okay, if our body required the fats, this acetyl-CoA is withdrawn from the respiratory pathway and form the water. Fatty acids combined with the glycerol and form the water fats. If our body requires the water proteins, then it's withdrawn from the pyruvic acid or acetyl CoA, withdrawn from the pyruvic acid or acetyl CoA and form the water amino acids, and amino acids combined form water proteins. Okay, means it's a making process. When our body requires the water proteins, when our body requires the fats, okay, these are withdrawn from the respiratory pathway and form the water fats or proteins. Means it's a making process. It's a making process. It's called what a anabolic process. Okay, it's called both steps are occurs in this ambibolic pathway, catabolism and anabolic. Okay, it's called ambibolic pathway. Okay, when this acetyl CoA is withdrawn, if our body requires the fats, then the acetyl CoA is withdrawn from the respiratory pathway and synthesize about fats. Okay, if our body requires the proteins, okay, these are from the respiratory pathway, pyruvic acid or acetyl CoA is withdrawn and form what are proteins. So both type of metabolism is occurs, catabolism and anabolism. That's why it's called what a ambibolic pathway. Okay, these are about a ambibolic pathway, and then RQ value. Both are completed. Both are important for 4 marks. Then next is important LAQ for 8 marks. C3 pathway or C3 plants. C3 pathway or C3 pathway or C3 cycle or C3 plants. C3 plants. Okay. Discovered by Melvin Calvin in a which plant? A green algae. This C3 pathway is discovered by which scientist? Melvin Calvin. Kelvin. That's why this cycle also called what? Kelvin cycle. That's why this cycle also called what? A Kelvin cycle. Okay. Discovered, experimented on which plant? Experimented on green algae. Green algae. Okay. Then the C3 pathway occurs in mesophyll cell of the leaf. C3 cycle occurs in mesophyll cell of the leaf. C3 cycle occurs in the mesophyll cell of the leaf, and the C3 cycle occurs in almost dicot plants. All dicot plants. C3 cycle occurs in almost all dicot plants. Almost all dicot plants. Okay. Now the C3 cycle occurs in how many steps? The C3 cycle occurs in 3 steps. C3 cycle occurs in how many steps? Occurs in 3 steps. C3 cycle occurs in 3 steps. Step 1, carboxylation. Step 1 is carboxylation. Step 2, reduction. Step 2 is reduction. Step 3, regeneration. Step 3 is regeneration. Okay. In C3 cycle occurs in how many steps? 3 steps. Carboxylation, reduction, and regeneration. Carboxylation means nothing but in this step, fix the carbon dioxide. Reduction means nothing but here, reduce the carbon dioxide. Reduce the Reduce the CO2. Then regeneration means again regenerate the RuBP. In this step, regenerate the RuBP means ribulose 1,5 bisphosphate is regenerate. Okay, so totally 3 steps are there in the C3 cycle. Step 1 is carboxylation. In this step, fix the atmospheric carbon dioxide. In this step, fix the atmospheric carbon dioxide. In this step, reduction is occurs. Okay, and regeneration means in the regeneration step again regenerate the what RuBP. Okay, now we discuss one by one. First, carboxylation. First step is carboxylation. And one more important point here, first stable product means when RuBP, okay, take the water atmospheric carbon dioxide, uptake the water atmospheric CO2, and first product is 3 carbon product is formed, that is phosphoglyceric acid, that's why it's called C3. These are 3 carbon compound. RuBP combined with the means it's take the water atmospheric carbon dioxide, and first stable product is formed is a 3 carbon compound. The name is phosphoglyceric acid. 3 carbon compound is a phosphoglyceric acid. That's why this plant is called C3 plant. Why it's called C3 plant? So first stable product is a 3 carbon compound. First stable product is a 3 carbon compound. That's why it's called what a C3 plants. Okay. Now first step is carboxylation. Okay. Step 1. Carboxylation. Okay. In carboxylation, what happened? 6 molecules of RuBP. 6 molecules of RuBP. What is the full form of the RuBP? Ribulose 1,5 bisphosphate. Ribulose 1,5 bisphosphate means ribulose have how many carbons? It has 5 carbon compound. Means like this. These are first carbon. These are second carbon. These are third carbon. These are fourth carbon, and these are fifth carbon. Okay. These are 5 carbon compound. Among the 5 carbon compound, first carbon and a fifth carbon. These are the first carbon and these are the fifth carbon have what a phosphate. That's why their position. This number gives the what a in which position I have what a phosphate. This number indicates the what a which position I have what a phosphate. Which positions have phosphate? First carbon and a fifth carbon. So how many phosphates are there? 2 phosphates. That's why we write the what a bis. Okay. Now RuBP accept the water 6 molecules of atmospheric CO2. 6 molecules of atmospheric CO2, and in the presence of Rubisco enzyme. Okay, Rubisco means same. Ribulose 1,5 bisphosphate carboxylase, and form a water 12 molecules of phosphoglyceric acid. 12 molecules of phosphoglyceric acid. These are 3 carbon compound. Okay, these are 3 carbon compound. That's why it's called what a C3 cycle. Okay, so in the step 1, what happened? RuBP accept the atmospheric CO2 and form a water in the presence of Rubisco enzyme. Product is 12 molecules of phosphoglyceric acid. These are the 3 carbon compound. That's why first stable product is 3 carbon compound. That's why this plant is called C3 plants, are also called C3 pathway. Okay. Here RuBP is a 5 carbon compound. Na? These are carbon. How many carbon? 6 carbon. Na? These are 5 carbon compound. How many molecules? 6 molecules. So 5 into 6. How many carbons? 30 carbons. Okay. These are 5 carbon compound, 6 molecules. Five carbon compound, 6 molecules. So 6 into 5 equal to 30 carbons. Okay. Here how many carbons? 6 carbons. 6 carbons. 30 plus 6 equal to 36 carbons. Okay. Now here these are 3 carbon compound and molecules are 12 molecules. So 12 into 3 equal to 36 carbon. Okay. Right. The next one. These are the step 1. These are the step 1. Carboxylation. Step 1 carboxylation, right? Then step 2. Step 2 is what? Reduction. Step 2 is reduction. In the reduction I have 2 sub-steps. In the reduction I have 2 sub-steps. Second step is reduction. In the reduction I have 2 sub-steps. First step 1 is phosphorylation. Step 1 is phosphorylation. Step 2 is actual reduction. These are what? Actual reduction. Okay. Step 1. Substep 1. In the reduction, substep 1 is what? Phosphorylation. Say here how many molecules of phosphoglyceric acid formed? 12 molecules of phosphoglyceric acid is formed. That's why here take the water. 12 molecules of phosphoglyceric acid. 12 molecules of phosphoglyceric 12 molecules of phosphoglyceric acid. Okay, it's utilize the take the 12 molecules of ATP. 12 molecules of ATP in the presence of phosphoglycerokinase. In the presence of phosphoglycerokinase. Okay. So how many molecules of this one is formed a product 12 molecules of 1, 3 phosphoglyceric acid is formed. 12 molecules of 1, 3 phosphoglyceric 12 molecules of 1, 3 phosphoglyceric acid is formed. Okay. See here. Phosphoglyceric acid 3 carbon compound, you know very well. Phosphoglyceric acid is 3 carbon. 1 carbon, second carbon, third carbon. So already at the third carbon I have what? Phosphate. Now this incoming phosphate, okay, ATP means 3 phosphate acid class. Okay, out of 3 phosphate, 2 phosphates are removed, means 12 ADP is removed. So how many phosphates are there? Only 1 phosphate. This phosphate is attached to the first carbon. First carbon. So first carbon and third carbon. First carbon and third carbon. These are the first carbon, these are the third carbon. Okay, in the first position and third position I have what? Phosphate. How many phosphate? 2 phosphates. That's why 1, 3 bis phosphoglyceric acid. Bis phosphoglyceric acid is formed. Okay, these are what? Phosphorylation. This step is called what? Phosphorylation. Then reduction. The reduction is what? 12 molecules of. The next step is reduction. 12 molecules of 1, 3 bis phospho. 12 molecules of 1, 3 bis phosphoglyceric. 12 molecules of 1, 3 bis phosphoglyceric acid in the presence of NADPH2. Okay, it's converted into the water. 12 molecules of phosphoglyceraldehyde, in short form PGAL. Okay, PGAL means phosphoglyceraldehyde. Phosphoglyceraldehyde is formed. Okay, 12 molecules of phosphoglyceraldehyde is formed and NADPH is converted to what? NADP. NADPH is converted to what? NADP. How many NADP? 12 NADPs. 12 NADPs. Okay, these are what? Reduction. So step 1 and step 2 is over. Step 1 is carboxylation and step 2 is what? Reduction. In the step 2 I have what? Again 2 substeps. First step is what? Phosphorylation. First step is what? Phosphorylation. Second step is what? Reduction. In the phosphorylation, how many ADPs are utilized? 12 ADPs. And in the reduction, how many NADPH are utilized? 12 NADPH. Remember this, 12 ADPs for conversion of phosphoglyceric acid to 1,3-bis phosphoglyceric acid. Then 1,3-bis phosphoglyceric acid to 12 molecules of phosphoglyceric acid is required the water 12 NADPH2. Okay, then next, last step is regeneration. Last step is regeneration. Regeneration. Which one is regenerated? RuBP is regenerated. Okay. So here this 12 molecules of PgAl, 12 molecules of phosphoglyceraldehyde, right? Individually 2 molecules 6 times. Right? Individually 2 molecules are 6 times. Okay. So 2 PgAl, then 2 PgAl, then 2 PgAl. Okay. Then 2 PgAl, then 2 PgAl, then 2 PgAl. Okay. 2 plus 2, 4, 4 plus 2, 6, 6 plus 2, 8, 8 plus 2, 10, 10 plus 2, 12. Okay, 12 molecules of PgAls, right? Individually 2 times. Okay. So 2 molecules 6 times. Now out of 12 PgAls, this PgAl is export into the what? Cytoplasm of the chloroplast. First, this PgAl undergoes first isomerase. Undergoes isomerase and converted to a dihydroxyacetone phosphate. Dihydroxyacetone phosphate. Okay. This dihydroxyacetone phosphate export into cytoplasm of the chloroplast. Export into the cytoplasm of the chloroplast and synthesize the hexose. Nothing but glucose. Hexose is a 6 carbon compound. It's called what? Glucose. Okay. So out of 12 molecules of phosphoglyceraldehyde, 2 molecules synthesize the water hexose. These last 2 molecules in the presence of isomerase enzyme convert DHAP. This DHAP export into the cytoplasm of the chloroplast and then synthesize the water hexose. Clear? So remaining out of 12, last PgAl synthesize the hexose. Remaining how many PgAls are there? 10 PgAls. 2 plus 2, 4 plus 2, 6 plus 2, 8 plus 2, 10. 10 PgAls are regenerate the RuBP. Out of 12, 10 PgAl regenerate the RuBP. Okay. Then this PgAl again undergoes isomerase. Okay, and convert to a DHAP. Okay, these all are 3 carbon compounds. All are 3 carbon compounds. Okay, now this DHAP condense or combined with the phosphoglyceraldehyde. This phosphoglyceraldehyde 3 carbon compound, DHAP also 3 carbon compound, and form 3 plus 3 6 carbon compound of fructose 1,6-bis phosphate is formed. Okay, 6 carbon compound of fructose 1,6 base phosphate is formed. Okay, in the next step what happened? See, fructose have 6 carbon now. So first carbon, second carbon, third carbon, fourth carbon, fifth carbon, sixth carbon. Okay, In the first carbon means this carbon and sixth carbon, last carbon, phosphate and a phosphate is present. Okay, 1,6 means their position, carbon position. First carbon and the sixth carbon have what a phosphate. Now, among the two phosphate, first phosphate is lost. The next step of first phosphate is lost. This step is called dephosphorylation. Okay. Enzyme name is fructose 1,6-bisphosphatase. Write in short form, phosphatase. Phosphatase, fructose 1,6-bisphosphatase enzyme. And from the first carbon, phosphate is last now. Last. Though, so product is what? Fructose 6-phosphate only. Fructose 6-phosphate. Fructose 6-phosphate. Okay. This step is called what? Dephosphorylation. This step is called dephosphorylation. Okay. Now, again, this fructose 6-phosphate combined with the PGAL. These are 6 carbon compound. These are 3 carbon compound. 6 plus 3, 9. Okay. And first condense and immediately cleavage. First condense and immediately cleavage into the 4 carbon compound and 5 carbon compound. 4 plus 5, 9 now. Here 6 plus 3, fructose is 6 carbon compound. PGAL is a 3 carbon compound. So these two are what? First condense. These two, 3 carbon compound and 6 carbon compound, first are condensed and immediately cleavage into the what? 5 carbon compound of xylulose. Okay, all are 2 molecules. Xylulose 5-phosphate, 5 carbon compound of xylulose 5-phosphate, and 2 molecules of 4 carbon compound of erythrose 4-phosphate. Erythrose 4-phosphate. Okay. Now, see, these are later involved. This one xylulose. First, this one. Erythrose again condense with the PGAL. Erythrose again condense with the PGAL. Erythrose is a 4 carbon compound. These are PGAL is a 3 carbon compound. 4 plus 3, 7. 7 carbon compound of pseudo heptilose or heptilose 1,7-bisphosphate. Pseudo P silent here. Pseudo heptilose 1,7-bisphosphate. Pseudo heptilose 1,7-bisphosphate. After condensation of erythrose 4-phosphate and PGAL. Erythrose 4-phosphate is a 4 carbon compound and PGAL is a 3 carbon compound. So this 4 carbon compound plus 3 carbon compound. 4 plus 3, 7 carbon compound. Both are combined now. So this pseudo heptilose is a 7 carbon compound. Okay. So here again 2 phosphates are there. 2 phosphates are there in the first position and 7th position. 1, 2, 3, 4, 5, 6, again 7. So first position has phosphate and 7th position has a phosphate. Okay. So from 2 phosphate, in the next step what happened? From the first position phosphate is lost. From the first position phosphate is lost in the presence of pseudo heptilose bisphosphatase. In the presence of pseudo heptilose bisphosphatase. Gem name is pseudo heptilose bisphosphatase. Pseudo heptilose bisphosphatase. Now product is which product? Heptilose 7-phosphate. Product is heptilose 7-phosphate. Heptilose 7-phosphate. Okay. Now this 7 carbon compound of heptilose, then 3 carbon compound of heptilose. 3 carbon compound of PGAL. Both are fused. 7 plus 3, 10 carbon compound. So immediately cleavage into the water 5 carbon and 5 carbon compound. Heptilose is 7 carbon compound. This is heptilose 7 carbon compound of heptilose and 3 carbon compound of PgAl. Both are fused. 7 plus 3 equal 10. So immediately cleavage, after condensation, immediately cleavage into the 2 molecules of xylulose, 2 molecules of xylulose 5-phosphate, 2 molecules of xylulose 5-phosphate, 5-phosphate, and 2 molecules of ribose, 5-phosphate, 5-phosphate, 5-phosphate. Okay, now this xylulose and this xylulose 2 plus 2, these 2 molecules of xylulose and these 2 molecules of xylulose undergoes which reaction? Epimerase reaction, epimerase reaction, and this ribose undergoes isomerase reaction. Okay, xylulose undergoes epimerase reaction and ribose undergoes isomerase reaction and form a what? How many molecules? Ribose 2 molecules plus xylulose 2 molecule, 2 plus 2 4, plus this xylulose 2 molecule, 4 plus 2 6. 6 molecules of ribulose, 6 molecules of ribulose 5-phosphate, 6 molecules of ribulose 5-phosphate. Okay, now again these 6 molecules are ribulose 5-phosphate. Utilize the water 6 ATPs and remove the water 6 ADPs. So already at the 5th position, ribulose have how many carbons? 5 carbons now. 1, 2, 3, 4, 5. Already at the 5th position have what? A phosphate. Like these are the 5th position, have phosphate. Now this incoming phosphate, like this 3 phosphate now. Out of 3 phosphate, TP, triphosphate. Out of triphosphate, diphosphates are removed. 2 phosphate, diphosphates are removed. Only 1 phosphate is there now. That phosphate is attached at the first carbon. Okay, so how many phosphates are there in which positions? 2 phosphates are there in which position? First position and 5th position. Okay, how many phosphates? 2 phosphate in first position and 5th position. So product is 6 molecules of ribulose 1,5. 6 molecules of ribulose 1,5 bisphosphate. Okay, 6 molecules of ribulose 1,5 bisphosphate. These are the 5 carbon compound. So regenerate the RuBP is regenerate. This total step is called what? A regeneration. This total step is called what? A regeneration. Now conclusion. How many ATPs, how many NADPH are required for complete Kelvin cycle? How many ATPs and how many NADPH2 are required in complete Kelvin cycle? Say a conversion of PGA, phosphoglyceric acid to 1,3 bis PGA. How many ATPs are? 6 ATPs are required, right? 6 ATPs are required. So here 6 ATPs, sorry, 12 ATPs, not 6, 12 ATPs, because of 12 molecules of PGA, na? Converted 12 molecules of 1,3 bis PGA. So here 12 molecules of ATPs are required, and in this step 6 molecules of ATP. So 12 plus 6 18 ATP molecules are required. Okay, conversion of phosphoglyceric acid to 1,3 bis phosphoglyceric acid, 12 ATPs are required, and then conversion of ribulose 5-phosphate to ribulose 1,5 bisphosphate. Ribulose 1,5 bisphosphate, how many ATPs? 6 ATPs. So 12 plus 6, 18 ATPs are required. 18 ATPs are required. And how many NADPH2? 12 NADPH2 are required. 12 NADPH2 are required. So one complete Kelvin cycle required the 18 ATPs and 12 NADPH2. Okay. So for one carbon fixation, how many ATPs are required? For one carbon fixation, 3 ATPs are required and 2 NADPH2 are required. Okay. For one carbon fixation, how many ATPs are? 3 ATPs 3 ATPs and 2 NADPH2 are required. Okay, these are complete Kelvin cycle. The next, okay, so again revise this. So a Kelvin cycle occurs in the mesophyll cell of the leaf. Okay, and then this Kelvin cycle occurs almost all dicot plants, right? So Kelvin cycle occurs in 3 steps: carboxylation, reduction, regeneration. Carboxylation means in the presence of RuBP co. RuBP co full form is ribulose 1,5 bisphosphate carboxylase. In the presence of ribulose 1,5 bisphosphate carboxylase, except the word atmospheric carbon dioxide, and productive, which product is formed? Phosphoglyceric acid, 3 carbon compound product is formed. Okay, that's why it's called what a C3 cycle. Okay, this step is called carboxylation. Okay, in the carboxylation, carbon dioxide is fixed. In the reduction, ATPs are utilized and then undergoes reduction step also. Reduction, 12 NADPH2 is converted to 12 NADP. Okay, then next regeneration. So in the reduction 12 ATPs are required, in regeneration 6 ATPs are required. Okay, and no required in the regeneration, NADPH are not required in the regeneration. NADPH2 are required only in the reduction step only. Okay, then next one, next is C4 plants. Tomorrow we discuss the C4 plants, general introduction of C4 plants. So C4 plants have some special characters. Okay, C4 plants can tolerate, means can stand in high temperature. Okay, and next is what the C4 plants have what a large biomass. It's a what a large biomass, and it's lost the water in less amount and minimize water. It's accept the water maximum carbon dioxide and loss the water, minimum water loss is lost. And this C4 plants have what a special anatomy. Which anatomy? Leaf anatomy. Here vascular bundles are surrounded by the bundle sheath cell. You know vascular bundle, xylem and phloem. For example, these are the phloem. Okay, now these are the xylem. Okay, phloem, xylem. It's called what a vascular bundle. This vascular bundle is surrounded by the bundle sheath cell. This type of anatomy does not seen in any plants. Okay, bundle sheath, vascular bundles are enclosed by the bundle sheath cell. It's called what a Cranch anatomy. It's called Cranch anatomy. Cranch anatomy means vascular bundles are surrounded by the bundle sheath cell. These are the word special character in the C4 plants. Okay, and these bundle sheath cells are agranular. Means what? Means generally C3 cycle occurs in only mesophyll cell. C3 cycle occurs only in mesophyll cell. But C4 plants or C4 cycle occurs in 2 cells. First one is mesophyll cell and second one is what? Bundle sheath cell. Okay, C4 cycle occurs in 2 cells. Bundle sheath cell and mesophyll cell. Okay, C4 plants are C4 cycle. C4 cycle occurs in 2 cells. Just today I'll give the introduction only, not in detail. Okay, C4 cycle occurs in 2 cells. First one is mesophyll cell. Second one is what? Bundle sheath cell. Okay, mesophyll cell have what? A chloroplast has a grana. Mesophyll cell chloroplast has a grana. Okay, that's why it's called what? A granular chloroplast. It's a granular chloroplast. Granular chloroplast. Then this bundle sheath cell, chloroplast is there now. This chloroplast, does not have what a granus like this. Does not have what a granus. Does not have what a granus. That's why it's called what? Agranular chloroplast. Bundle sheath cells are agranular chloroplast. Okay, two types of chloroplast present in the C4 plant. First one is granular chloroplast in the mesophyll cell, and second one is agranular chloroplast in the bundle sheath cell. That's why chloroplast shows the dimorphism. Di means two. Morphism means morphological structure. Okay, so bundle sheath cell does not have chloroplast. Mesophyll cell have what? Has a chloroplast. Two types of chloroplast is there. That's why it's called what? A dimorphism chloroplast. Okay, these are the special character. And cranch. What is the meaning of cranch? Cranch is nothing but in the English vritta. Vritta means nothing circular. Nothing but circular means these bundle sheath cells. These are the bundle sheath cell. These bundle sheath cell enclose the vascular bundles. Vascular bundles are nothing but xylem and phloem. Okay, and tomorrow we discuss in detail the C4 plants. Okay.